Hi friends, welcome to Blessed Excellence Center. Do well to subscribe to our channel. In our last video, we looked at the preliminary step of de novo synthesis of purine nucleotides. Today we are going to look at the main steps of purine de novo synthesis, part 1. Because of the lengthy nature of it, I have to divide it into two parts. So this is part 1. Now, you see this stuff here, G square Tg dot arrow dot caft dot W. This is a summary of the purine de novo synthesis. It summarizes the 10 steps of purine de novo synthesis. Your presenter remains E.B. Christian. Now, let's look at some keynotes on that you need to know about the purine de novo synthesis. There are 10 main steps of purine nucleotide de novo synthesis. This is summarized in the acronym G square Tg arrow caft W. Now, the G here, first G stands for glutamine, second G stands for glycine, T stands for tetrahydrofolate. This G stands for glutamine. The arrow stands for ring closure. C stands for carbon dioxide. A stands for aspartate. F stands for fumarate. T stands for tetrahydrofolate. W stands for water. So the summary of it is that in the 10 steps, for first step, glutamine is used. First step uses glutamine. The second step uses glycine. That is, it involves incorporation of glycine. Third step uses tetrahydrofolate. Fourth step uses glutamine. Fifth step does not use any precursor. It is the step where the ring is closed. That is the first ring closure step. Good. The sixth step, carbon 4 oxide is incorporated. Step 7, aspartate is incorporated. Step 8, fumarate is released. Nothing is incorporated, rather there is release of fumarate. Step 9, tetrahydrofolate is inculcated. Step 10, nothing is inculcated, but water is released. At the same time, ring is closed. That's ring closure. Now, it's important you note that step 2, step 4, step 5, step 6, and step 7 require ATP. And a biochemical reaction that requires ATP is usually driven by a group of enzymes called synthetase. Okay? So this is like a summary, or it captures the entire steps. I'm going to be very slow. I'm going to detail you with structure how to understand and know the 10 steps of the purine nucleotides de novo synthesis. In this video, I shall be doing 5 steps. Then the next video, we shall be giving attention to the remaining 5 steps. Now let's look at the first step. The first step of purine de novo synthesis involves the addition of amino group. When you see NH2, it's called amino group. So the step involves addition of amino group to the product of preliminary step. Remember, in the preliminary step, we formed PROPP, that is phosphoribosyl pyrophosphate. Phosphoribosyl pyrophosphate. You remember how this guy was formed? I told you that in purine de novo synthesis, we use ribose 5-phosphate that is obtained from hexose monophosphate shunts. Okay, so it is upon ribose 5-phosphate that we now build the purine base. Okay, good. So in the reaction, we use ATP. We use we pull out 2 phosphate group from ATP and attach it to ribose 5-phosphates, and that leads to the formation of this phosphoribosyl pyrophosphate, which is the product of the preliminary step. It is now the substrate of the first step. So we are saying here that the first step involves the addition of amino group to this guy called phosphoribosyl pyrophosphate with the corresponding removal of pyrophosphate. Remember that when you have two phosphate group together, it is called pyrophosphate. So when you attach the amino group, you will remove the pyrophosphate, and then that will lead to the formation of 5-phosphoribosyl beta amine. Now it is the amine that is attached is obtained from glutamine. That's why I said that glutamine is the source of the amino group. After adding, it becomes glutamate as driven by glutamine PRPP amidinotransferase. So the point now is that in step 1, the amino group is added to phosphoribosyl pyrophosphates, and then pyrophosphate group is removed. When you remove it, the enzyme that drives the reaction is called glutamine. Glutamine phosphoribosyl pyrophosphate amidinotransferase. Look at the pathway here. In this reaction, you can remember the structure very well in the previous video when we talked of the preliminary step of purine de novo synthesis. This is what was formed. We formed this compound. So this is what we call phosphoribosyl pyrophosphates. Now to this phosphoribosyl pyrophosphate, you add glutamine. When you add glutamine to it, the work of the glutamine is that the glutamine will donate amino group that will replace the phosphate group. So you pull out this phosphate group and then add amino group here. Then the pyrophosphate group you pull out is now removed this way, eliminated this way. Then water is incorporated. We use water to cut or to cleave or to detach the amino group from the glutamine so that it can be added here. And if you remove amino group from glutamine, it becomes what? Glutamate. So this is the first step. The structure is very simple. This is just phosphate group, one phosphate group, and this is two phosphate group, and this is ribose sugar, and this is phosphate group, this ribose sugar, and this amino group, and that is what we call 5-phosphoribosyl beta amine, which is the first step. The enzyme of the reaction, as I've said earlier, is called glutamine phosphoribosyl pyrophosphate amidinotransferase. Now let's look at the second step of the reaction. The second step is where phosphoribosyl beta amine condenses with glycine to form glycinamide ribonucleotide GAR. The reaction requires ATP and as such catalyzed by GAR synthetase. Look at the pathway here. Look at the reaction here. You can remember this very well. You see, one thing that makes biochemical pathway easier is that the product of the previous step becomes the substrate of the next step. So this is the product of the first step, and it is now the substrate of this second step. Just repeat it, and you'll discover that what you'll be doing small things to it, and you're having the product. That's what makes the pathway easier. Now look at the structure for glycine. In this second step, this glycine is added to phosphoribosyl beta amine. Look at the nature of the reaction. In the reaction, this carboxylic group will be attached to this amino group. Okay, good. And the nature of the reaction is that if you pull out hydroxyl group from this carboxylic group, you pull one hydrogen from here, water is formed, and then you attach this, the carbon oxygen CH2 and NH2 is what we are seeing here. This is carbon oxygen CH2 N2 here in NH2. Okay, and then you remove one hydrogen here to form water, and you are now left with this. So that's what is attached. Every other component is repeated. That is how simple the pathway is. All you need to just pick the whole of these components, attach it here, and then eliminate your water. So we call this glycinamide ribonucleotide, GA, and it requires ATP. So since it requires ATP, we call it GA synthetase, the enzyme that drives the reaction. Now let's look at the third step. Third step is the step where formyl group CHO is added to glycinamide ribonucleotide, forming formyl glycinamide ribonucleotide, FGA. It is a formylation reaction and is driven by GA transformylase, which does not require what? ATP. I told you in our previous video about some terminologies associated with purine de novo synthesis, and one of them is formylation reaction. And I say that formylation reaction is a type of reaction that involves the addition of formyl group, that is aldehyde group, to a substance. Now this stuff you're seeing here is the product of the second step, so you repeat it exactly the way it is without any alteration. Now the only difference will be small alteration. Here you have amino group, so to this amino group you add formyl group, to obtain this. So you see that this is NH2, and then when you add the formyl group, become NH, and then this is formyl group that is added here. That's why I use different color to differentiate, so you see the points of reaction, the point of attachments, and every other component is repeated. The phosphate, the ribose sugar, the other group of the compound, the other group are repeated. Only to this amino group you add formyl group, and when you do that, you form formyl glycinamide ribonucleotide. So you see how the name is still derived from here. This one is glycinamide ribonucleotide. So when you add formyl group to it, it becomes formyl glycinamide ribonucleotide. And since it involves addition of formyl group, the enzyme that catalyzes the reaction is called transformylase. And since you are adding the formyl group to GA, to this GA here being glycinamide ribonucleotide, the enzyme will now be GA transformylase. And where do we obtain the formyl group we added? It is obtained from N10 formyl tetrahydrofolate. And when this N10 formyl tetrahydrofolate donates the formyl group, it becomes tetrahydrofolate. And that is why you see the arrow illustrating. So that is the steps, or what happens in the third step of de novo synthesis of purine. And let's go to the fourth step. This step involves the addition of amino group from glutamine to formyl glycinamide ribonucleotide, yielding formyl glycinamidine ribonucleotide. The reaction requires ATP and as such catalyzed by FGAM synthetase. Note, water is added to detach amino group from glutamine. You can remember one of the steps where glutamine was added. In fact, the first step, glutamine was added. So this is the second step where glutamine is added. Look at the reaction here. The same thing. Look at this. This is the product of third step. This is the product of the third step. We just repeat it the way it is, and we call that formyl glycinamide ribonucleotide. Now, to this you add glutamine. When you add glutamine, the work of the glutamine is to donate amino group. When it donates the amino group, it becomes glutamates, and then ATP is required to drive the reaction. We use water to detach the amino group from the glutamine to form glutamates. Okay, so if you add, look at this, you have carbon bonding oxygen. That is the point where you attach the amino group. We have it here. If you attach the amino group here, you have it here. And that is the only difference, the point of attachments. If you observe very well, this compound and this compound are looking almost similar. This is the secret that makes the pathway and the structure easier. You observe that you'll be repeating the whole thing the same way it is with small differences. And that makes it easier for you guys. Okay, good. So when you attach the amino group here, you have it here this way, and it becomes formylglycinamidine. When the amino group has not been added, it is called formylglycinamide ribonucleotide. But when you attach the amino group, it becomes formylglycinamidine ribonucleotide. And since it involves an ATP, the enzyme that will drive the reaction is called F-jam synthetase. That is this guy, this product, this formylglycinamidine ribonucleotide synthetase. That is the fourth step. Now let's go to the fifth step. The fifth step does not require the incorporation of any precursor, but involves ring closure. To achieve this, ATP is needed. So in this step, formylglycinamidine ribonucleotide undergoes ring closure to form 5-aminoimidazole ribonucleotide as driven by a synthetase. Now look at this reaction here, the pathway here. In this step, you don't need to add anything. You don't add any precursor. You only close the ring. Look, I have, how many atoms are here? You have 1, 2, 3, 4, 5, and then for this one, 6. So you use this compound here to form imidazole. In Chem 361, you were taught of imidazole. Imidazole is actually a pentagon with 2 double bonds and 2 nitrogen atoms. So use the whole of this to form the imidazole. Now watch how you form it. This is the phosphate group. The phosphate group, we don't touch it, we repeat the way it is. Ribose sugar, the ribose sugar, we repeat it, okay? Good. Now this nitrogen here is this nitrogen. Is linked to this carbon, which is this carbon you're seeing here, okay? Now there is amino group here, which is this amino group you're seeing here, okay? Good. This CH2 is this CH, okay? Then this amino group, this nitrogen is this nitrogen. Then this C is this C. Now here is CH2, but here is C. That is what brought about this double bond you are seeing here. So you can see this is a 5-sided shape with 2 nitrogen atoms, and we call that imidazole. So it has amino group attached to it at position 5. If you start from to count, this is 1, 2, 3, 4, and 5. So we call this 5-amino imidazole ribonucleotide. That's the name of this product. So in the fifth step, no precursor is inculcated, but there is ring closure. And since it requires energy, you call the enzyme synthetase, and it is from what is formed that will be the synthetase, because it means the formation of this product, which is 5-aminoimidazole. So we call it 5-aminoimidazole synthetase. That is a synthetase. So that is what happens in the fifth step. Thanks for watching. Do it to subscribe to the channel, share the video, click on the bell, like it, and as well drop your comments.